First up this morning will be the Biosystems Division, talking about their work with microbe-host environment interactions. And I'd like to introduce Professor Ina Hoschel here, and she'll be starting us off. Are we good?
Yeah, thank you. Good morning. So my background and research area is statistical and quantitative genetics. Statistical genetics is just a branch of biostatistics. And it obviously strongly overlaps or encompasses quantitative, population, evolutionary genetics, and it can be viewed as statistical genomics and bioinformatics with the added value of genetics. A particular area of interest at the present time for statistical geneticists is systems genetics or genetical systems biology, which is a field that essentially merges the problems and methods from genetics, systems biology, and genomics, and has been described as the next frontier in systems biology and medicine. The current main focus of my research is on developing and applying statistical methodology for the interpretation of large-scale multi-omics human datasets. Okay. So here we have a diagram of the influence of genetic variation or DNA sequence variation, which of course influences gene expression or mRNA levels, subsequently protein levels and structure. Proteins function together to determine events at the microphysiological level, flow of metabolites within and across biochemical pathways, which then in turn dictate events at a more macrophysiological level, which then influences subclinical phenotypes, which eventually lead to clinical phenotypes. So as an example, we have cardiovascular disease, and subclinical phenotypes for cardiovascular disease are measures of atherosclerosis or plaque accumulation in the arteries, which can be measured using ultrasound imaging techniques. And the influence is not just from top to bottom, but we now know that environmental factors and risk factors, more generally risk factors, for instance, body mass index, right, is a risk factor for CVD, cardiovascular disease, that these risk factors influence mRNA levels through epigenetics, for example, DNA methylation. And in the past, essentially all we have done in these so-called genome-wide association studies has been to associate DNA sequence variation with the clinical phenotypes. And these studies require national and international consortia and thousands to 10,000s of individuals, so they are expensive and difficult to organize. And although they certainly are continuing to make important contributions, today we associate DNA sequence variation with all levels of this hierarchy. So they are expression SNPs, they are protein SNPs, they are methylation SNPs, they are SNPs influencing the subclinical phenotypes, they are SNPs influencing risk factors such as BMI, and typically the effects of these SNPs on all of these intermediate phenotypes are much larger and much easier to detect than the effects on clinical phenotypes. So I consider myself very fortunate to be the lead statistical collaborator for several multi-omics projects that are based on the multi-ethnic study of atherosclerosis, or MESA. MESA is a prospective cohort with almost 7,000 participants from 4 races or ethnicities. And the phenotypes measured in MESA are all related to atherosclerosis. So mostly different measures of plaque accumulation. Of course, there's also a collection of extensive clinical, social demographic, lifestyle, medication, nutrition, whatever data. And these data have been collected at baseline in 2000, I believe, and at several subsequent exams. Exam 6 is planned for next year. I forgot to mention that MESA, my collaborators are at Wake Forest Medical School in Winston-Salem, which is one of the founding institutions of MESA. And in MESA, genome-wide SNP data are long available. And the data that we have currently added to MESA at one particular exam, exam 5, genome-wide data on now approaching 2,000 individuals, including gene expression, DNA methylation, and microRNA at the present time. Of course, some limited proteomics and some other things. So this study and this comprehensive data set allows us to really study this hierarchy. So what we have done initially, of course, is to associate gene expression with the atherosclerosis phenotypes, to associate DNA methylation with the atherosclerosis phenotypes, to associate, of course, in detail, to investigate the association between methylation and gene expression, to associate risk factors with methylation and gene expression, to perform mediation analyses of the effect of DNA methylation, which is mediated by gene expression on the subclinical phenotypes, to perform mediation analysis of the effect of risk factors, which is mediated through methylation and gene expression on the subclinical phenotypes, to associate DNA sequence variation with expression, with methylation, with subclinical phenotypes, with risk factors. And as I mentioned before, we have done this at one particular exam, so this has been essentially a cross-sectional study. We are now expanding the study into a longitudinal study, pending funding, because MESA has data at different exams and continues to plan future exams. And we will also focus more going from association and mediation analyses to prediction of disease. So one particular interest that I currently have is to use the DNA sequence variation to perform causal analyses in this data set. So randomization is very well known to be the most convincing method for establishing a causal relationship between any two variables. And Mendelian randomization uses randomization of alleles during meiosis, which of course precedes the effect on a molecular or clinical phenotype, gene expression, atherosclerosis. So Mendelian randomization uses the randomization of alleles during meiosis to perform causality tests that are less susceptible to hidden confounding. MESA is an observational study, so confounding is of course a big issue. Much of the early Mendelian randomization work has focused on estimating the effect of a modifiable risk factor such as BMI on disease outcome, CVD, using a single SNP. My interest is on network, something that I call network Mendelian randomization, which uses multiple SNPs, other external variables, essentially to compare different, to not perform mediation analysis conditional on a certain causal model, but to compare different causal models using structural equation modeling. So in particular, we are interested in our data to identify those genes for which this causal chain holds, right? We have a risk factor such as BMI that influences, leads to changes in DNA methylation, which leads to changes in gene expression, which leads to changes in atherosclerosis. And to identify such a causal chain is not trivial because, first of all, there is the potential for hidden confounding for any of these links. There may be a confounder between methylation and gene expression or gene expression and atherosclerosis. And there's the potential for reverse causation for any of these links. Gene expression can be responding to a change in the subclinical phenotype. And of course, there are cases where gene expression actually regulates methylation instead of the other way around. So we think that this type of Mendelian randomization analysis, causal analysis, could be which we are currently exploring in detail, could be an important step between these types of genome-wide omics association studies and clinical trials. Thank you.
I'm Alan Dickerman. How do I move to the next slide? Kayaking the fire hose of information. And I, like Ina, am in the Biosystems division and don't do lab work myself. I just analyze data. And I want to just give you a flavor of some of the sort of case studies or just examples, fun examples of data that I've been working on in the last year that were that I've found interesting, and some of it might spark some interest in some of you. On the both of these slides or figures represent a kind of molecular clock analysis that is a computationally intensive. There's a Markov chain Monte Carlo. It's a resampling of the likelihoods of different trees that can fit the phylogenetically organized data. On the left, we have porcine epidemic diarrheal virus. That's a serious challenge to the swine rearing agriculture, and it's swept into North America in about the last 6 years. And we have collaborators in X.J. Meng's lab who is interested in the analysis of this virus in the American pig industry, and well, it's pretty obvious that it came from Asia because that's where it was rampant shortly before it came here, and they wanted to put a date on the time of arrival of PEDV into North America, and they had sequenced a strain of the virus, and there were 2 other existing sequences of the virus from isolates in North America. And there were several complete genomes of the virus from samples spanning a number of years in Asia. And this small blue square represents the isolates in North America, and you can see that they've got a very short phylogenetic distance among them. And knowing the dates from several of the isolates in Asia, we could put an estimate of, I think the estimate was for the spike, the peak of the blue, the probability distribution of the time of the divergence of the American strains was about 3 years. Now that's the divergence among the ones that are in America. So the arrival into America was the ancestor of those. So that is a lower bound on the time of arrival, and the upper bound on arrival was that next age group, which is about 7 years. That next peak has a bound of around 7 years. So between 7 and 3 years is the time of arrival of this virus into the pig population in North America. Then I'm working on a similar analysis in mosquito vectors of malaria, the Anopheles genus. And there's an interesting story on the divergence of New and Old World Anopheles, and I'm trying to see what the correlation is with the biogeographic, the separation of the continents and the opening of the Atlantic Ocean. I suspect that we can't pin the separation of New and Old World Anopheles on opening of the Atlantic Ocean. It's subsequent to that, but it's interesting. Another whole area that I really like to work in is gut microbial diversity analysis, and people sometimes call this metagenomics, microbial metagenomics. I think real microbial metagenomics should be limited to when you're sequencing genomic DNA from around the whole genome. These kinds of studies typically focus on amplifying small subunit ribosomal RNA, so you're not analyzing the whole genome, you're analyzing a marker, but a very informative marker that you can put into a taxonomic context with some level of precision. And these particular data are from a horse gut microbiome in a study where they were using a probiotic to see what shifts in the microbial community you observe. And in this particular plot, I mashed together a few different things. You can plot the isolated strains or samples along with, and you can't read them, but some of those labels are from full genomes pulled out of GenBank. So and put them on a phylogenetic tree. I think that's as close to nailing down the relationships of your sampled small subunit ribosomal RNA sequences to, quote, known genomes. That's as fine a resolution as you can get, but it's too fine, so it's not suitable for summary back to publish because you've got, say, 500,000 different branches on your tree, and you can't visualize a tree that way. So you usually end up putting it into pie charts and stuff like that. Over on the right, this, quote, heat map is showing, well, the two axes on it are samples and not taxa, but biochemical functions enriched or present in the taxa. So that was an attempt to go from samples and changes over time to biochemical potential of the microbiomes involved. Just kind of running through these because I'm sampling a bunch of different kinds of studies. This is another, I have to say that the basis of all of this that I do is the capability of doing very, very dense, very high throughput, but high throughput isn't really the word, but large volume data production. And this is showing microRNAs mapped onto the genome. And just in general, analyzing microRNAs is another great field. It's really interesting, and you can always find new things. And in this case, I'm showing a novel microRNA in the mouse genome that we're working on. And finally, this is a new project that I'm doing with my wife, Rebecca Wadham, and she's got a collaborator at St. Jude's Hospital, St. Jude's Children's Hospital, and this is analyzing presence or absence of different strains of Staphylococcus in this case. Staphylococcus, presence or absence of genes in those genomes associated with disease phenotypes in the patients that they occur in. And we're trying to do a statistical analysis of the association of genes in bacteria with phenotypic outcome in the patients. And we really need Ina's insight on that. Next.
So I also am playing timer today, and I feel really awful about it because I'm hearing all this exciting research, but I'm having to show 2-minute warning. So please excuse that throughout the day. So we can start with the focus of my research program, which is on human fungal symbiosis. And ultimately, we're not just interested in the molecular interactions, but the coevolution of these interactions that have transpired over what we consider millions of years, and ultimately how the environment, past, present, and potentially future, is impacting these environments. So we utilize a very clinically relevant fungi, Aspergillus fumigatus, which is also, to a certain extent, a model fungus to work with. And so as you're all breathing in right now, don't be alarmed, but you're inhaling fungal spores as we speak. Very low doses, but chances are all of us will be fine by the end of the day and throughout our lives because we have these healthy immune systems. In the evolutionary arms race that's transpired, again, over millions of years, we tend to be the victor. And what we're really wanting to understand in the laboratory is, well, how do we maintain this immunity, so to speak, against this fungal pathogen? And so Aspergillus, again, is really interesting because it encompasses this host microbe environmental cycle, because you'll get, in a specific subset of hosts under certain environmental predispositions, you'll get an invasive form of the infection. And this occurs in the umbrella term of immunocompromised individuals. We're specifically interested in the mechanistic understanding of these different forms of immune suppression and how they lead to disease. As you can see there, the different disease phenotypes are associated with very high mortality rates, in some cases up to 90% in certain subset clinical populations. Now, what's even more interesting in my eyes is that this same fungus, in a completely same host, but under different environmental constraints, you can have this hypersensitive response occurring. So in this case, you'd have roughly 193 million individuals who suffer from asthma each year, cases. And of this 5 million will develop this completely opposite spectrum form of disease phenotype, which is this ABPA. So again, hypersensitive response. And about 500,000 individuals will end up developing a more severe form. So again, Aspergillus biology, to me, is very unique because you have this exposure, and in the vast majority of cases, you'll see this immunity. But in individuals where you have this immunocompromised phenotypes, you'll see infection, and another subset of the population, you'll see allergy. So again, it's really this environmental constraints that's playing an important role in disease manifestation. So we have, through a lot of collaborations, we have two different, paradigms that really look into the models. One is this host-centric phenotype, where we really look at host susceptibility factors. So we're able to put in different environmental inputs, and these can be anything from steroid treatments to neutrophil depletion to chemical agents that are associated with treating other disease phenotypes, because Aspergillus, again, is an opportunistic fungal pathogen. We can input a variety of fungal strains, both of clinical origin, and we really utilize a cadre of different techniques, measuring fungal burden, survival studies. We've been working on the immune response with NIML, which we're really grateful for, and some large-scale transcriptomics analyses. And so what we're ultimately wanting to understand is that when we use this umbrella term of having an immunocompromised host, it really isn't the case. It's a very broad term, and when we start looking at the specific instances or types of immunocompromisation, then you start getting mechanistic insight into, well, what's really going on here that's facilitating an environment that allows the fungus to grow. And so that's really been prevalent in our transcriptomics data that I'm not going to touch on too much here, but what we're seeing is that depending on the host phenotype, you're providing different avenues for the fungus to thrive and proliferate. And what's really important here is that if you are creating next generation of antifungals, they might not necessarily be ideal for each type of immunocompromised suppression system. So again, specificity, or I'm using this term kind of loosely, personalized medicine is really central for the next generation of therapies. So we also focus on more of the pathogen side, and this is an offshoot of my PhD work where we identified these effector proteins from really important plant pathogenic fungi and oomycetes. And so this concept of these effectors manipulating host environments is very well understood in the plant microbe community and in certain communities associated with bacterial infections of mammals. But in the fungal community, it's been incredibly challenging to identify these effectors. So using a mixture of computational sciences and wet lab sciences, and again, this slide usually represents a 1-hour talk I give, we really try to go iterate back and forth between integrating and analyzing and processing a diverse source of information. It may be transcriptomics data or genomics. We do a large-scale comparative genomics. And then we iterate this in the wet lab because we have these predictions, but unless we can validate them and identify physiological relevance using our murine and mouse models or some really detailed biochemistry and molecular biology, we can start making some conclusion and answers to what we're doing. And so ultimately, what we're wanting to look at is, can we take these subsets or different tools that have been created by individuals and really dissect the contributions of the host, the environment, and the microbe. And so again, we're using genomics, biochemistry, and a lot of computational sciences. Because ultimately what we want to understand, and I'm reiterating this again, is the impact of the environment on producing different types of disease phenotypes, and in certain cases, none at all. And only when we have this mechanistic insight into disease manifestation can we then utilize this to create novel translational therapies. And so I've tried to keep that in my time frame, and I think, all right, didn't go off. So we can have the panel session now, and we'd be happy to take questions. I'm also going to ask if you have questions, there's a microphone up here, and to please come up here and ask your questions. Thank you.
How can biology mesh with statistics? It's unreasonable to ask them to sequence 10,000 genomes. They can't do it. So I'm wondering how you deal with that in your own research when there are indications that they are not statistically sound, to give them, you just throw it all out, or?
I've always been a little paranoid about the statisticians telling me that the sample size is too small. These days, I don't know if this is on. Is this working? Anyway.
It's not working.
These days, we can get much bigger samples, but I would like to ask Ina, what is big enough? And I would also like to ask Ina if she thinks that her study with almost 7,000 patients, is that big enough?
Well, I'm honestly a little surprised. Our studies always begin with planning and an NIH proposal, and the first thing we do is sample size calculations, and it's totally clear then what we can do. I think any study should start with sample size calculations, then you don't have these surprises. And I can't believe that they wouldn't start that way. So everything we...
They do. So from the biological standpoint, I think that when you're submitting a grant to NIH and from a biology standpoint and you say, I'm going to sequence all of these people, from a biological standpoint, 200 is a lot, but I guess okay, sorry.
Well, I think part of it is I'm used to collaborating with medical schools whose background is GWAS, where sample sizes are in the thousands to 10,000, so they're used to large sample sizes. And these concerns are almost foreign to me. One thing is that you shouldn't be doing classical sample size calculations. You should be doing sample size calculations that are adequate for genomics. I see people do classical sample size calculations based on criteria that lead to unnecessarily large sample sizes. And sometimes you need to do sample size calculations based using numerical methods instead of classical simple formulas.
Maybe you can talk later.
But I think to start out with sample size calculation is the most important thing that you can do.
Sorry, as a biologist, that seems that my understanding is that there's no any way we're going to make statisticians happy. But I think the size of the sample maybe depends on the biological question, right? 200 sounds to me like an obscene number. Thinking of how much money I'm going to pay for those 200 samples. So maybe I can see that the larger the sample, the better and more accurate the predictions would be. But maybe if we limit the question, then that would be a way to limit the size of the sample. Am I correct on this approach?
Absolutely
So
true. You have to ask the right questions and maybe do pilot studies to see what effects you're dealing with. Yes.
So I wanted to ask a question to everybody on the panel about the computation analysis and the challenges you face, because there's some interest in our group, of course, and want to find a way to connect between the kinds of questions you had. And just one quick elaboration on what was answered just now. Do you try also to build out a theoretical framework first so that then you decide what sort of data you collect, or you first collect the data usually and go the other way around? And this is a question that I've always been intrigued about.
If you don't mind, I'll start this one. So in a perfect world, I think we try to collect as much data as possible. So some of our transcriptomic studies, we have these 10 different environmental constraints that we can utilize, but including some baseline responses, but we really have to pick, what's most relevant for funding purposes or for our various stakeholders, right? So we can't necessarily cover the whole gamut, so we really have to be choosy. And I think those constraints are relevant. Initially, I think with a lot of these things, someone who's kind of, in my example, gone from the plant side to the human side, we're very fortunate that there's more of a network architecture in regards to molecular interactions. And I think a lot of groups are utilizing this solid foundation either through protein-protein interaction networks or KEGG and reactome pathways as real foundational resources. However, I think that's just the beginning of something that could massively build over a period of time. And I think what we are doing and what others are looking at is, we're looking at comparative genomics. So you're seeing all these different fungi and you're seeing all these different sequences, but you're seeing a large degree of similarity also. So then not just looking at it from a genomic standpoint, but then integrating the transcriptomics as well. So which genes are necessarily being turned on, at what level, at what time point are they being turned on? And so to really understand the mechanistic insights, you need a high resolution of data. And to be able to integrate that into a model is, I think, the real challenge and where most groups are entering. I think last question from Silke.
Another question about samples and data, because it seems to me that very often there would be other useful data sets out there that could help you in your work apart from your own ones. But very often, again, it seems to me, I don't have much experience on that, is they are very difficult to access. Or they are very difficult to integrate. Yes. So is that true, and what could be done to maybe help them?
A lot of hard work. I've trolled and mined through the SRA, the short or sequence read archive, and brought out data sets to compare to Illumina sequence data sets that have been produced in the DAC here, or in the core lab here. And that can be done, but it's always difficult, but it can be done. It's computationally, to address the previous question, it's computationally challenging, but it's really great to be in a well computationally supported institute and have just downloading those data sets can be like hours. So it's amazing, but we can do it, but it's never easy.
In humans, I think it's maybe less, or in medical studies, it's maybe less of an issue because, first of all, we always have to replicate our results in other cohorts. And since these cohorts are NIH funded, they must deposit the data. Mm-hmm. So for us, it's never an issue to find data sets, unless it's something that just nobody has done. And computation is a challenge, but so far, everything we do, we are able to do with the resources here at Wake Forest.
I think I'll spare my answer so that we give enough time for the biology fellows to have their nice talk. So with that, thank Alan and Ina for their presentations this morning. Thank you so much.